Hello, everyone. My name is Lucas Koester, and I work in the Department of Veterinary Microbiology and Preventive Medicine at Iowa State University in the United States. Today, I would like to talk about mining the U's vaginal microbiota using 16S rRNA gene amplicon sequencing to identify bacterial phylotypes associated with sheep reproductive performance. To give you a brief overview, I will start by talking about some terminology, then introduce our study, talk about our methods, and present our results. Then we will have some discussion and concluding remarks, followed by our future directions. Let me take a moment to introduce and clarify some microbial ecology terminology. The microbiota are all of the microbial taxa in an environment. In this case, the ovine vaginal microbiota. An amplicon is a segment of DNA that is amplified, in this case, the bacteria-specific 16S rRNA gene. An operational taxonomic unit, or OTU, is a cluster of sequences based on sequence similarity. We will be using an OTU as the basic unit of comparison between the different treatment groups in this study. Alpha diversity measurements are values taken from a single sample, such as species richness, which is the total number of different species, species evenness, which is how evenly the abundance of each organism is distributed, and species diversity, which is a weighted combination of the two mentioned before. And finally, beta diversity is a comparison of these measurements between samples. Here is a quick cartoon example using fruit to describe the alpha diversity measurements, species richness, species evenness, and species diversity. The first community on the far left only has 3 types of fruit, and the apple accounts for a majority of the community. This would be an example of low species richness, only 3 types, and low species evenness, as the fruits are not evenly distributed, favoring the apple. The second community from the left has a low species richness with only 3 species, but the community is much more evenly distributed. The final community has a high species richness and species evenness. And as species diversity is calculated using both species richness and species evenness, the higher these values are, the higher the species diversity as well. So, why should we study the microbial communities within these systems? Well, they could be used as indicators or predictors for a disease. They could be directly causing the disease. And finally, they could be controlled to prevent a disease or ameliorate the disease's effect. So we wanted to know if we could identify microbial phylotypes within the ovine vagina that could act as indicators for reproductive performance, specifically relating to establishing and maintaining pregnancy. There are 2 objectives in this pilot study. Objective 1 was to compare sheep that successfully conceived during breeding to those that did not. And objective 2 was to monitor healthy ovine vaginal microbial communities throughout a successful pregnancy. To accomplish this, we swabbed the vaginal microbial communities from 14 Hampshire and Hampshire Suffolk crossbred ewe lambs at 3 separate time points. S1, which was taken pre-breeding, S2, which was taken at the time of pregnancy checking, which is about 2 months after breeding, and S3, which was taken directly prior to parturition. The flock used in this pilot study had a history of poor reproductive performance, so we ensured that the breeding rams underwent a breeding soundness exam prior to breeding. For objective 1, we swabbed all 14 animals pre-breeding to capture the microbial communities that may interfere with conception. Of the 14 animals in the pilot study, only 4 ewes successfully conceived. As I had mentioned before, This particular flock had a history of poor reproductive performance. Nevertheless, we were able to compare the pre-breeding vaginal microbial communities between those animals that successfully established pregnancy to those that did not. For objective 2, we monitored the vaginal communities of the 4 ewes that became pregnant to get an idea of what a healthy vaginal microbiota consists of throughout pregnancy. DNA from vaginal swabs taken during the pilot study was extracted using the Qiagen Powerlyzer Power Soil Kit, which includes a rigorous mechanical lysis step. We then amplified the 16S rRNA gene using 250 base pair paired end aluminum-MySeq sequencing chemistry. Sequences were quality filtered, organized into OTUs, and classified using Mother. And finally, data handling as well as graphics generation were completed using the R package phylotypes. Now, on to the results. We will start with the pre-breeding comparison between pregnant and non-pregnant ewes. Initially, when comparing the average relative abundance of the 20 most abundant OTUs, we noticed some striking differences between treatment groups. Specifically, the abundance of families and genera that contained known pathogens seemed especially high in the non-pregnant treatment group. OTU1 classified within the gram-negative Pasteurellacea family, which are known to contain lipopolysaccharide, an endotoxin capable of destroying endothelial cells. This family includes Mannheimia hemolytica, known to cause pneumonia, and Pasteurella multocida, which has been shown to infect the reproductive organs causing tissue necrosis. OTU3 was classified within the Ureaplasma genera, which have been isolated from ewes with a history of infertility. OTU5 was classified as a Histophilus species, which has been associated with several disease symptoms, including infertility and ram-lamb epididymitis. OTU7 was classified as a Staphylococcus species, some of whom are causative agents for bacterial vaginitis. OTUs 8 and 9 were both classified within the Corynebacteriaceae family, which contains some species known to cause caseous lymphadenitis. This can lead to severe damage to the reproductive organs. And finally, OTU 18 was classified as Campylobacter species. Campylobacter jejuni and Campylobacter fetus fetus are both known to cause abortions in sheep. These all seemed more abundant in ewes that were unable to conceive, although they showed no obvious symptoms. So we did a little digging, and we found that the average relative abundance for each treatment group was not representative of the individuals within the treatment populations. This is a heat map showing the relative abundance of the 20 most abundant OTUs per ewe, faceted by treatment group. We noticed that the individual ewes usually had high abundance of a single potential pathogenic OTU, thus shifting the group average. Nearly all of these potential pathogens were represented in the non-pregnant animals at the pre-breeding time point, however. Additionally, we wanted to see how the treatments compared on an entire community level. This is a principal coordinates analysis where we can compare each ewe's microbial community to the others in the study. Each point represents a single sample's entire microbial community, and the distance between points represents how similar it is to the others. In this case, the greater the difference, the larger the distance. We ultimately discovered no discernible differences between non-pregnant and pregnant animals for the pre-breeding time point, which was surprising given the seemingly major shifts in the highly abundant OTUs. But this isn't entirely unrealistic, because besides the large increases in the abundance for the individual pathogens within individual samples, the rest of the microbial community is quite similar. In summary, We found a high abundance of different potential pathogens in ewe lambs that did not conceive during breeding. These potential pathogens were only found within certain individuals, skewing the group average. Additionally, no differences were detected between the two treatment groups when considering entire microbial communities, despite the observed increases in potential pathogen abundance. Now we will switch to Objective 2, analysis of the pregnant animal throughout pregnancy. The goal of this objective was to characterize and compare the ovine vaginal microbial communities of pregnant ewes over the pregnancy interval. We began by comparing the alpha diversity measures species richness, species evenness, and total species diversity between sample time points S1, S2, and S3. Compared to the other time points, S2 reported lower species richness and diversity than either S1 or S3. This suggests a lower number of different species, and it suggests that certain species within this community tend to dominate, making the community uneven. These differences can be attributed to a few possible factors, including weather patterns or the pregnancy stage itself. A similar story is seen when comparing the entire microbial communities between time points. Once again, each dot represents an individual sample's entire community. They are then colored by the time point that they were taken in to distinguish between treatment groups. We see that there is a possible difference between time points S2 and S3 and very distinct clustering occurring within sampling time point. We also compared the relative abundance of each phyla between individuals faceted by time point. This data shows a large increase in fusobacteria and proteobacteria within individuals from the S2 time point, as well as a decrease in Bacteroidetes when compared to the other two time points. We also noticed that the time point S1 is the only time point with the phylum Tenericutes represented. Finally, the S1 and S3 sample time point communities also seem to be more consistent across animals. In summary, throughout a successful pregnancy, shifts in the entire microbial community as well as alpha diversity measures were detected between sample time points, especially when comparing the second time point, S2, to both S1 and S3. Additionally, regardless of the shifts in microbial populations, both the ewe lamb and the offspring were considered healthy at the end of the study. In conclusion, or to summarize our pilot study, several potential pathogens found in pre-breeding ewe vaginal microbial communities may play a role in causing infertility, and these organisms may utilize different mechanisms to achieve the same effect. Additionally, none of the non-pregnant animals showed any classical symptoms of these different diseases, yet they all did not conceive. And with this pilot study, we identified shifts in vaginal microbial communities of pregnant ewe lambs over the course of their pregnancy. These shifts did not compromise the health of the ewe lamb or the offspring in any case. Moving forward, we have conducted a trial with a much larger sample pool, with 68 animals of similar genotyping and farm conditions. Once again, all rams underwent breeding soundness exam prior to breeding, and we swabbed the penile sheath of the breeding rams to potentially identify a source of infection. With the increase in sample size, we hope to improve our statistical power and more accurately detect the organisms that may prevent conception. We also hope that this research will provide fundamental knowledge about how these microbial communities interact with the host and that we can apply this information to increase reproductive success in a production farm setting. With that, I would like to thank you all for listening to my presentation. I would also like to thank Dr. Youngs and Dr. Schmitzhesser for the opportunity to work on this project. I would like to thank our farm manager, Mr. Trey Jackson, as well as the Iowa Sheep and Wool Board for funding this project. And then, of course, I would like to thank the organizers of the Third World Conference on Sheep for the opportunity to speak. Thank you, and I will field any questions now.